So in general, complement pathways always tend to be a topic that's very overwhelming in the sense that there's a lot of different steps. And we're not going to go through, every detail and every cofactor, but it is important to just have kind of a general understanding of this pathway and then, what factors we should be familiar with. So I'm going to go into a little bit of the detail and then we'll kind of zoom out and look at this big picture to try and understand, what do we really want to take away from this? And, and there are situations where you might get questions that are more detailed, but in general, you don't have to know every single detail. You just have to have an idea as to what's going on. Okay. And so that's essentially what we're going to try to kind of include here. And the thing that I want you to keep in mind as we're going through this, what are the things that you really have to know? Again, we really have to know these functional components of complement. Okay, that's the big thing. So if you had to take one thing away from all of this and you're watching this and you're like, I'm not really sure what I want to know. Number one is I want to know the functional components of complement and what they each do. Okay, so C3A, C4A, we talked about these being anaphylotoxins involved in anaphylaxis. When I say anaphylaxis, we're referring to histamine release from mast cells, right? C5A also does that, but it also stimulates neutrophil chemotaxis, which is particularly high yield. The neutrophil chemotaxis point, remember, we're getting neutrophils to the site of inflammation. C3b, very much involved with opsonization. We talked about that. And the other thing that we talked about was IgG, right? IgG, also very good at opsonization. Okay. And then C5b to C9, right? We talked about membrane attack complex, basically perforating holes into the cell membrane. So those are all the big things to keep in mind, and you'll see them all come into play as we're going through these pathways. All right, let's start by talking about the classical pathway. Okay, so we're just going to kind of go through this one. Now, if you understand this pathway, the other pathways are actually pretty straightforward. Okay, so this is going to be kind of the steepest learning curve, but once we get through this one, the other ones will make a lot of sense, I think. So the first step, we're going to have to bind to our microbe. Okay, so this is our microbe. It's our pathogen. It could be a bacteria, it could be a virus, whatever. And on this microbe, you can see there's some IgG antibody. Now remember, IgG antibodies at the Fc portion, like we talked about in the previous videos, particularly that Fc portion that's closer to the hinge region on the antibody can bind complement. And what is it binding specifically? It's binding specifically C1. Now remember, it's not just IgG that can bind complement. It could be IgG or it could be IgM. IgM having more binding sites in the pentameric form can make it more likely for it to bind complement. So just another thing to kind of keep in mind when you're talking about, the structure of some of these immunoglobulins. But again, the point here, IgG is on the surface, it's tagging this microbe, right? So here it's binding to the epitope. We have our Fc region portion down here, complement's binding to that, right near the hinge region, and we can see C1 is the component that's binding to it. Okay, so that's this C1 complex. Now this C1 complex, what it does, and remember what I said, we start at 1 and we end at 9, right? So it doesn't mean that we go necessarily in the correct order as we're going through the steps, but we do start at 1 and we end at 9, so that's something to keep in mind. So C1 complex is going to split C4 and C2. Okay, so you can see C4 here, you can see C2 here. It's essentially splitting them. And so one thing that I want to make kind of clear here with complement, if we start with something, let's just say we started with C4, and if we split it, when I say we split it, what am I talking about? I'm talking about splitting into two components, okay? So you're going to get a C4A, okay? So you're going to get a C4A, and then you're also going to get a C4B, okay? So you're going to get a C4A, and you're going to get a C4B. You can think of this kind of subletter as being more of an activated form, whereas more of the inactivated form you can think of as just kind of having the number. Okay, so this being more inactivated and these being my activated forms. Now we already know what C4A does, right? C4A, we already said, stimulates histamine release, right? It's one of the anaphylotoxins. C4B though, what does that do? C4B is going to go down into the next step, and the same thing is true for C2B. Okay, so both of these, all of the B's are going to stay in the complement cascade. Now, in the past, there was different notation for this, where the C2A was the component that stayed in the complement cascade, but kind of the updated way this is categorized is if it's going to be one of the B components, so if it's a C4B, if it's a C2B, if it's a C3B, that component can stay in the complement cascade, and that's the way to remember it. So if you get the B component, right, if it has the B here, it's probably, in some ways, possible that it's going to stay in the complement cascade. So if you have a B component, right, it's most likely going to be in the complement cascade here. Now C3B has some additional effects like opsonization, okay? But in general, these B components can stay in the complement cascade. This microbe marked with an antibody, the C1 complement factor bound to the Fc portion on the IgG antibody, or it could have been IgM, and then that cleaved the C4 and C2 complement factors to C4b and C2b. Those come together and now they form a C3 convertase. So the convertase is telling you what this structure does. It takes C3, right? It would take a C3, just like we did with our C4, and it's going to cleave it into two components. Again, this C3 convertase is made up of C4b and C2b, okay, on the surface of this microbe. And you can see that happening here. So now we're going to, once we form this C3 convertase, okay, so C4b2b forms a C3 convertase. Once we form this, now a C3 can be coming by and we cleave that C3 into two components, just like we talked about. So we have a C3a. Okay, so C3a, we already know what that does, right? We set up here, functional components. C3a is also going to be involved with histamine release from mast cells. And the B component, we just said, the B component sticks around. Okay, so the B component comes down into the next step and helps form the C5 convertase. Okay, now the C5 convertase, it's like it says, once again, the C5 convertase is going to convert C5. Okay, and again, this is formed now from C4b, C2b, and C3b. Okay, so this is kind of where it gets confusing. But again, it's telling you what it does, right? So it's going to cleave C5. Now, once we finally get to this step where we form this C5 convertase, we're able to cleave C5. And again, we get our two components. We get C5a, which we already know what this does, right? We go up to our functional components here, right? C5a, we know that it's going to stimulate histamine release from mast cells. It's also going to be involved with neutrophil chemotaxis, right? So that's what C5a is going to be doing. And then we know C5b, it has a B in its name, so it's going to kind of continue this cascade. And we know we're home free because if we remember, the membrane attack complex, what is that made up of, right? We just talked about this. It's made up of C5b to C9, okay? C5b to C9. And so this C5b component is going to combine with C6 through C9, right? So they're all going to come together and they're going to form the membrane attack complex, okay? So once we get the membrane attack complex, remember that's the terminal step of this complement cascade. Okay, there's really two kind of terminal steps that I think about. The first terminal step, like we talked about in our first video on this, was going to be C3b. C3b, it has a B in the name, right? We said if it has a B in the name, it can continue down this cascade. So C3b can continue to form the C5 convertase, right, in the next step, or the C3b can then go on to stimulate opsonization. So the C3b can do either one, right? That's kind of the first terminal point of this cascade is opsonization from C3b formation. But the second terminal point of this cascade is going to be the membrane attack complex in the sense that this is a structure that isn't going to go on to another step, and it's a structure that can punch holes into this microbe's membrane. Then we're getting into the common pathway, right? And that essentially we're saying common because all of these different pathways, right, they're going to have some slight differences up here, but once you get to this C5b step, you can see they all have C5b here, you're essentially going to be going down the same pathway, right? You get to C5b, the C5b is going to bind to C6 through C9 and form the MAC. Okay, let's go through the lectin pathway here really quickly. So the lectin pathway, and the nice thing about this lectin pathway is it's essentially the same thing as the classic pathway. The only difference is in the first step. In the first step of the classical pathway, we had IgG or IgM antibodies bound to the microbe surface, and so we bound our C1 complement to that. But in this situation, instead, you're going to have mannose, right? You're going to use these mannose structures on the microbe surface, and you're going to have mannose binding lectin, right? Mannose binding lectin, MBL, is going to bind to the mannose, okay? And then that structure is going to kind of act as that initial C1 from the classical pathway. Every step after is going to be the same. This mannose binding lectin, just like C1, is going to cleave C4 and C2. The B's are going to stay. We know what the A's do up here. The B's are going to stay, so the B's are going to come together, and they're going to form a C3 convertase, which tells you what it does. It converts the two components of C3 to C3a, which again, we know what that does, right? It's one of our functional components. And then C3b can either go on to do opsonization or can go into this next step to form the C5 convertase. C5 convertase, it says what it does in the name. It converts C5, right, to C5b and C5a. C5b can combine with C6 and C9 to form the membrane attack complex. Go right back down the common pathway. So the only difference between the classical pathway and the lectin pathway is the first step. Okay, so that's where, if you're kind of thinking, okay, well, how would they ask me a question? Do I have to know all the details? you want to know the differences, right? So you want to know that the classical pathway is associated with immunoglobulins, and the lectin pathway is associated with mannose binding lectin, kind of triggering the cascade. The alternative pathway has to do with C3 starting things off. Okay, so it's a little bit trickier. So let's just go through that really quickly. So the first step has to do with C3 essentially binding to the cell surface to form C3b. Now, once you get C3b here on the cell surface, okay, which again is completely different from what we talked about with the other two pathways, there's also going to be a second component, a factor B. And when that factor B gets cleaved here, you end up forming a C3 convertase. Now, like I said, I'm not going through every single detail here because it's just not high yield, right? We don't need to know every single cofactor. But I am going into some detail because, this is one of those things where if you understand this a little bit, you can make a lot of sense of the disorders and see where some of this stuff comes from. So with the alternative pathway, it's a little bit harder because it doesn't have the same features as the other two pathways initially. Your C3 convertase is actually different. Okay, so if there was a question where they wanted you to distinguish what makes up a C3 convertase, right, you can see in the alternative pathway, it is completely different than in the classical and the lectin pathway. Okay, so this is made up of C3b and then this B factor sub B component. Okay, and then this eventually what it does is it acts just like the name says though. A C3 convertase is cleaving C3. And so you're going to get further C3 cleaved. You're going to make more C3b, right? That can go back into this pathway, just like in the previous steps, and form a C5 convertase. So again, after those first initial steps, everything, beyond the C3 convertase is the same once again. So if you understand everything beyond the C3 convertase pretty much, you can understand each of these pathways. It's just that the first step is a little different. So let's just go through the text here just to hammer this home. So again, when we're talking about the classical pathway, we're talking about immunoglobulins setting things off. Which immunoglobulin specifically? IgM or IgG. You get a C1 complex that can bind to the Fc portion, splitting it C4 and C2. When we split something, we get the A and B components, right? We know where a lot of these structures go. If it has a B in its name, though, it can stick around. Okay, so if it has a B in its name, it can stick around. So the C4b and 2b stick around. They form the C3 convertase. We cleave C3. We get the B component that sticks around. Now we form a C5 convertase, right? And that takes us right into the common pathway, where we'll then form C5a, C5b. The B sticks around, helps form the membrane attack complex. Okay, lectin pathway, same thing. The only difference is we activate with mannose, and we have mannose binding lectin that forms the C1-like complex because it does essentially what the C1 complex does. Everything else is the same as the classical pathway. Alternative pathway, the difference is we're activating on the microbial cell surface with C3b bound to the surface. It's going to bind this factor B, and then we're going to form a C3 convertase, eventually forming a C5 convertase once we cleave C3b and add that to our structure. Okay, and so again, we get to the common pathway. It's the same thing. They're all going to end in this membrane attack complex formation. Okay, so they're all going to eventually get to that step. They just start a little bit differently. Okay, so the C1 esterase inhibitor, and we'll talk about the relevancy of this with C1 esterase inhibitor deficiency, but the C1 esterase inhibitor is going to prevent this first step. So if I had some, let's just put C1 C1 inhibitor here, it would essentially inhibit what this C1 component is doing. And what is it doing? It's cleaving C4 and C2, right? So it's inhibiting this step. So you can imagine if you had too little of this, if you had a deficiency of this C1 esterase inhibitor, you wouldn't be able to stop C4 getting converted to C4b, C4a. You wouldn't be able to stop C2 being converted to C2a, C2b, right? So you would have a lot of complement activation in this initial step. You'd have very low levels of C4 because you wouldn't be able to inhibit or regulate it going into form C4b. Okay, so that's why low C4 levels is very classic for C1 esterase inhibitor deficiency for that reason, right? Because you can't inhibit this step. So this C4 is constantly getting converted, right? Not regulated. All right, the next one on here, the next inhibitor is going to be delay accelerating factor, also known as CD55. And we see a deficiency of this with PNH, paroxysmal nocturnal hemoglobinuria, which we'll talk about in a minute. And this essentially prevents the formation of the C3 convertase. Okay, so you can see, where's my C3 convertase? I have them here, right? So decay accelerating factor is going to inhibit this step, right? It's going to inhibit these two structures coming together to form the C3 convertase. In our body, right, in homeostasis, these molecules like decay accelerating factor, what they're doing is they're regulating things, right? So if you have too much decay accelerating factor, you're not going to be able to have complement mediated destruction. But if you have too little of it, you're going to have too much destruction, right? So that's the concept. Essentially, these are helping to regulate this process so we don't have overactivation. And the final one on here is the membrane inhibitor of reactive lysis, CD59, which is going to prevent the formation of the membrane attack complex. So in all of these, right, it's going to combine with C6 through C9, and this is going to make our MAC, right, which is essentially going to be C5b through C9. And what this CD59 is going to do is it's essentially going to inhibit at this step. Okay, I'll just put MIRL here for membrane inhibitor of reactive lysis. Okay, so that's what that stands for. And again, it kind of looks like the MAC name, and that's maybe one way to remember it. And it's essentially inhibiting this step, preventing the formation of the membrane attack complex.